I'd like to return to our discussion of the kingdom Euplantae today. And when we classify the true plants, then it's really important. One of the important characteristics is looking at modifications to the plant alternation of generations. And this is something we discussed at the end of the last module, and I would encourage you to review that before continuing on with the discussion of the Euplantae. So again, we'll talk about some modifications in this alternation of generations today, but that is one of the things that we consider when we're classifying the true plants. Now, another important characteristic of how we classify the true plants will be whether or not they have so-called vascular tissues. And so the vascular tissues are of two types. There's xylem and phloem. And xylem is a tissue that conducts water primarily from roots to leaves, and I'll discuss that in a minute. And phloem is a plant tissue that conducts sugars primarily from leaves to roots, although really sugars can be conducted anywhere in the plant. And again, the phloem will carry out that. So some plants have these vascular tissues and other plants do not. And the advantage of having the vascular tissues is that you can move large volumes of water and also sugars long distances. And so this allows plants to be bigger for one, and it gives them a little bit of an advantage and distances them from some of the more aquatic areas or more humid areas. But we'll come back to that later. So when we look at the arrangement of vascular tissues, there are certain possibilities. We find vascular tissues in roots, stems, and leaves, and the arrangement of the vascular tissues really defines what part of the plant we're looking at. So a root isn't necessarily just below ground, for example. Stems are not necessarily just above ground. Stems and roots sometimes you'll see roots above ground, sometimes stems below ground. So it's the arrangement of the vascular tissues that defines that. So in roots, we tend to see the vascular tissues in the center of the root in what we call a vascular cylinder. In stems, we'll see the vascular tissues arranged around the periphery of the stem, either in bundles or sometimes in rings, as we see in trees. And in leaves, most of the time, it's a linear arrangement of the vascular tissues. So the veins of a leaf then would be the vascular tissues. So again, kind of following up on that then, as we look at a cross-section of an actual root, again, here's where the vascular tissues would be. And when we look closely at that, we see the xylem and the phloem. So in a stem, again, we'll see the vascular tissues arranged in bundles around the periphery of the stem. Or they can be scattered. This would be another characteristic of stems. Also, the vascular tissues can be scattered throughout the stem, and this is an example from a corn plant. And then also in woody plants, the vascular tissues will be arranged in rings. So when we look at wood is composed primarily of xylem. In fact, it is composed of xylem. And most of the mass of a tree is actually xylem. And then again, the veins of a leaf also would be composed of the vascular tissues, xylem and phloem. Veins of a leaf. Okay, so another thing I want to mention about the vascular tissues is if we look at xylem then, xylem is a kind of tissue that we describe as being lignified. So in addition to the cellulose that we find in cell walls, also these cell walls will contain this material lignin. And lignin is a hard, relatively rigid material. And lignin is what gives wood its woody texture. And so again, looking more closely at xylem, xylem is actually a dead tissue. As lignin is deposited in the cell walls, it's relatively water impermeable, and so that actually kills the cells. And so what we end up with then are perforated tubules that connect to one another. And so this allows water to move pretty freely through the cells. They're not impeded by any kind of a cytoplasm. And so we'll talk more about this in a couple minutes, but for example, through capillary action then, water can be drawn up through these tubules. So xylem is kind of interesting because again, it does lend some structural support to the plant, and it also is a dead tissue. So again, when you're looking at a tree, if you're looking at a pine tree or an aspen or something like that, most of that tissue that makes up that trunk is actually non-living material. And I mentioned earlier that most of the mass of the tree would be tied up in xylem, but really most of the mass of the tree is composed of water, which is inside the xylem. So again, xylem is involved primarily in conducting water from root to leaves, and we'll get back to that in a minute. So again, that woody stem then is mostly xylem, and again, containing a lot of water. So this process then of getting water from roots to leaf is known as transpiration. And this is a really important process for plants. So water then will enter the xylem in the roots. It will work its way up the stem and eventually be lost from the leaves. And so there are basically three kind of forces that are involved here. One is what we call root pressure. And so root pressure would be the osmotic pressure that draws water into the cytoplasm. So the cytoplasm of the roots is going to be hypertonic, hopefully hypertonic to the surrounding soil. And so water moves into the cells. Also, there is some capillary action here because cellulose in roots, in the cell wall of roots, acts kind of like a paper towel. So that will suck water into the roots as well. But we call this drawing of water in through osmotic pressure or possibly capillary action in the cell wall. We refer to that as root pressure. So roots will draw water in. And then once water enters the xylem vessels, then capillary action takes over. And so again, we get water into the xylem, and then the water can move up the xylem through capillary action. Also, again, there is some of that capillary action involved in the cellulose cell walls of the roots. And then water works its way up the xylem through the trunk. And then the third factor here is what we call evaporative pressure. And that's in the leaves. The leaves have pores associated with the surface of the leaves. And these are called guard cells, and the openings are called stomata. And so when the stomata are open, water will be lost from the plant. And this is also how carbon dioxide gets access to the inner part of the plant so that photosynthesis can take place. So water then is being lost through evaporative pressure by the leaves, and so that then removes some of the mass up above. So as water is lost from the leaves, that allows water to move up through capillary action and root pressure. So the important thing about these three forces working together, the root pressure, the capillary action, and the evaporative pressure, which is a negative pressure at the top, is that it really doesn't require energy input by the tree. These are really just natural processes: osmosis, capillary action, and then evaporation. So anyway, the plant then can draw in tremendous amounts of water, move that through the tree or move that through the plant, and then also nutrients can be brought in, limiting nutrients can be brought in. So transpiration is a really important process. And then, as I mentioned earlier, phloem is all about moving sugars. And so there are two types of cells that we find in phloem: sieve elements, which actually transport the sugars, and companion cells. And so companion cells are complete cells, nucleated cells. The sieve elements are living, but they lack nuclei, although they do have a cell membrane. And so again, here would be the sieve elements. And so the sugars would be moving through the sieve elements, and then the companion cells control movement of the sugars. And so together they form phloem. And so this movement of sugars we refer to as translocation. And so translocation of sugars is accomplished by what we call loading. We'll load sugars usually at the leaf. So sugars will be loaded into the phloem, typically through active transport. And so that will create a hypertonic solution. Then the sugars will follow their concentration gradient. And kind of at the other end, usually in the roots, we have what we call a sink, where the sugars are unloaded from the phloem. Again, this may be through active transport. And so sugars are removed in the root, they're loaded in the sugars. That creates a concentration gradient. The sugars will follow that. And also then, because the sugars are so concentrated at the source where we're loading them in, water will be drawn in there, and that will create a hydrostatic, a fluid pressure within the phloem, and that will also help move the sugars along, and we refer to that as bulk transport. So again, it's most typically from leaf to root, again, loading at a source, unloading at a sink, concentration gradient, and then bulk flow will move those sugars through the phloem. And that's a vital process, of course, because the roots, again, do have living cells. They need to live, but they're not above ground, so they can't photosynthesize. So those cells are going to need energy. They have to get that energy from sugars, and the sugars are being produced via sunlight, photosynthesis, the leaves, so the roots need to get that nutrition. And if something happens to prevent those sugars from being delivered to the roots are going to die, and eventually that's going to kill the tree or the plant, depending what kind of plant it is. All right, so as we look at the kingdom Euplantae then, there are several divisions that we want to consider, and the first of these is the division Bryophyta, which are the mosses. And the mosses are an avascular type of plant, meaning that they lack vascular tissues. And again, as we look at mosses, I'm not really sure if you've seen mosses before, but they are they look like they are vascular, but they're actually not. So they look like they have leaves, but these are not true leaves. They lack true roots. And the green leafy part of a moss, when you're looking at that, that's actually gametophyte tissue. And we'll come back and talk about that in a second. So again, they are avascular, meaning they lack xylem and phloem, and so they tend to be found in very wet habitats because fluids are going to have to diffuse from the surface of the moss into the interior of the moss. So they tend to be kind of thin and relatively small compared to vascular plants. Now, another characteristic is that they require water for fertilization. So water is required for fertilization in the mosses, which means that sperm are going to have to swim to fertilize an ovum. And then thirdly, as I mentioned, they are gametophyte dominant. So when we look at that life cycle, most of the life cycle of these plants is tied up in the gametophyte generation. So the gametophytes are physically larger, they live longer than the sporophyte plants will. And so again, as we look at this life cycle, we're going to see an alternation between a gametophyte plant and a sporophyte plant. And that also means then when you're looking at mosses, when you're looking at green, quote unquote leafy looking mosses, they're gametophytes, which means they're also haploid plants. So the haploid generation is the dominant generation. So again, by dominance I mean physically larger and longer lived. All right, so as we look at the life cycle here, again, we can kind of jump in anywhere, but what we'll do is we'll start off looking at the gametophyte plant. And again, the role of a gametophyte plant is to produce gametes. So again, the green leafy part of a moss would be the gametophyte. And if we look at gametophytes, at the top of gametophytes, this is where the gametes are going to be produced. And you may recall that gametes are sex cells, sperm or eggs. So again, we can kind of jump in here anywhere. So we'll start in with spores. Spores would be haploid cells. When spores begin to grow, they will grow into a gametophyte. And this is a slide of spores that are starting to divide mitotically. We call these baby gametophytes protonemata. So these protonemata are these individual filaments. Those are the product of a haploid spore. And each of these little filaments then can grow into a moss plant. So again, each of those protonemas can grow into this moss plant. And then that gametophyte plant's job, again, is to produce gametes. So if we look at the top of these sprigs, that's where gametes will be produced. And so some of those will produce antheridia. So an antheridium is a structure that produces sperm. So again, if we look at the top of some of these sprigs will produce antheridia. And so this is a slide of the top of a moss sprig. And we're looking at the antheridia, these kind of oval structures, and then the tiny granules inside are sperm. So all of this is haploid tissue. All of it is generated through mitotic division. So again, we're looking at individual antheridia here, and the tiny granules inside are sperm granules. And so the antheridia then will rupture, and the sperm will be released, and they need water because they want to swim and find an ovum and fertilize that. So the next part of this life cycle is then that on the top of some of the sprigs of moss, then there will be structures called archegonia, which produce ova. And so again, if we look at other sprigs of this moss, instead of antheridia at the tops, we will find archegonia. And so again, this is a slide looking at a longitudinal section through the top of one of these sprigs. This is the archegonium, and then this large cell inside is the ovum. And so the archegonium, it's a little hard to see from the structure, but the archegonia are a little bit like a vase. So if you kind of look at this vase that I have here on the table, there's actually an opening at the top. They'll be enlarged at some point, and that's where the ovum is found. So what will happen then is that spermatozoa that rupture from the antheridia will swim to an archegonium, and they'll swim down the top of the archegonium and fertilize the ovum, which is housed in this enlarged region down here. And so looking at our slide again, we can see part of this in our slide, but what happens again is that the spermatozoa will swim down the top and then fertilize the ovum here. And of course, when they fertilize the ovum, it's no longer an ovum, it's now a diploid zygote. So again, sperm will make their way down the top, fertilize the ovum, that forms a zygote. So the zygote then is housed within the archegonium at the top of one of the moss sprigs. And so the next phase then is that zygote will begin to mitotically divide and grow into a new plant. That plant is a sporophyte plant. And so in our picture here then, these are the sporophytes here. And so these sporophytes are growing out of what was an archegonium. They literally grow right out of the top of the archegonium. And then there will be this enlarged structure at the top known as a capsule, and the capsule will contain the sporangia. And sporangia are structures that make spores. So all of this is diploid tissue. So we consider this a separate plant. It's the process of a sexual process. Sperm swimming, fertilizing ova, creating a zygote. Zygote mitotically divides to generate the sporophyte plant. And so in the case of the mosses, the sporophyte is actually attached to the gametophyte. It grows right out of the top of the gametophyte. And these will only live for a couple of weeks. They dry out. Their main job as a sporophyte plant, phyte means plant, sporo means spore, is to produce spores. So the spores will be produced within this capsule up here. And the capsule contains sporangia, structures to make spores. So again, the fertilization will occur, zygote begins to divide, and the sporophyte is going to grow right out of the top of it. And then sporangia within the sporophyte will produce spores undergoing meiosis within those. So again, we're going to look at a slide that will actually be a section right through one of these capsules at the top of the sporophyte plant. These are the sporangia within. We can see the tiny granules inside. Those are the individual spores. And so the actual tissue here is diploid, but meiosis occurs within the sporangia, and these are haploid spores. So when we talk plants, spores are always going to be haploid. And then the tip of this capsule will break off. The spores are released. So again, there's the sporangia with the spores inside. And then the spores are released, and we get back to where we started. The spores will, if conditions are favorable, will grow into new gametophytes. So we tend to find mosses in fairly wet habitats. We find mosses throughout the Yellowstone ecosystem. Examples would be the juniper haircap moss. Another example is the wet rock moss, common beard moss, aspen moss, which we find growing on the trunks of aspens. So, and these are just a few. There are actually quite a few mosses that we find up in the Yellowstone ecosystem. Now, another relative of mosses are what we call liverworts, and we do encounter one particular liverwort up there pretty frequently. The liverwort takes its name from the fact that instead of having kind of a leafy-looking gametophyte, it has a more broad and flat type of gametophyte. And the one that I most typically see up there is what's known as the lung liverwort. Now, there are a number of other plants that are tracheophytes. And so the tracheophyte plants then would be plants that do have vascular tissues. So again, the mosses are not tracheophytes. They are avascular. And so the rest of the plants that we're going to talk about are tracheophytes. They possess vascular tissues. And all of the tracheophyte plants are sporophyte dominant. So the plant that you normally see, if it has any size to it at all, would be a sporophyte. So sporophyte dominant, meaning that the sporophyte will live a lot longer than the gametophyte, and it's physically larger than the gametophyte. So most of the plants that we're familiar with are tracheophytes and are sporophyte dominant. Now, one kind of plant that we see a lot of in the Yellowstone ecosystem, and you've probably seen many times as well, that is an example of a tracheophyte, are ferns. So this is the division Pterophyta, and the word ptero here means wing, but it's really referencing leaves. So the Pterophyta then are structurally complex. They grow in fronds, they're vascular, so they're complex plants, but they are, in terms of their life cycle, they are relatively simple. So again, I think everybody's familiar with ferns, but again, structurally very complex. They have true leaves, they have true roots, they have a true stem. But in terms of their life cycle, primitive. So they're tracheophytes. And so since they are relatively primitive in their life cycle, what that means is that they do require water for fertilization. So when I say plants require water for fertilization, what I am saying about them is that they do not produce seeds. So in our life cycle that we discussed just now in terms of the moss, I never mentioned a seed. So mosses don't produce seeds. But what you might also find interesting is that ferns are not seed-producing plants, even though they're a fairly common plant globally. And again, they're sporophyte dominant. So when you look at a fern, you're looking at a sporophyte plant. This is a diploid plant. That's the sporophyte. And again, the job of a sporophyte plant, phyta means plant, sporo means spores. The role of this plant then is to make spores. So there's a little bit of a twist in the life cycle here compared to what we talked about previously. But where we're going to start here is looking at the sporophyte. We want to look at how the sporophyte produces sporangia and eventually produces spores. So again, when we look at a fern, we're looking at the sporophyte plant, and here's some young ferns growing in the Yellowstone ecosystem. And what will happen here is on the lower surface, typically of the leaflets of the fern fronds, certain structures will develop. And these structures are known as sori. Sorus is singular, sori will be plural. And what a sorus is a cluster of sporangia. So again, the terms I'm going to be using here, one is sorus. Sorus means a cluster of sporangia. A sporangia or sporangium is a structure that produces spores. And so keep in mind that within the sporangia, meiosis is going to occur. So the sorus and the sporangia are diploid, and then the spores are going to be haploid. So I want to actually show you, what these sori look like and where these sporangia are. So I'm going to take a fern frond here. So as we look at this fern frond, these are the individual, leaflets of the fern frond. And you'll notice that there are these spots on the bottom of the leaf. And a lot of times students will, bring in ferns like this and sometimes ask me, and they'll say, oh, I think there's some kind of insect eggs on my fern. But no, this is part of their natural life cycle. So these little spots that we're seeing here, these are the sori. And again, the sori are composed of clusters of sporangia. So they usually start off kind of greenish, and then as they mature, they darken like that. So again, looking at this on a microscope, we're looking actually through the surface of a leaf here. And so again, this is our sorus. This big structure here would have been one of those brown spots that you saw on the leaflet. And if we look closer at that, then you'll see that there are these individual sporangia that make up the sorus. So the sorus is a cluster of sporangia. And then within each sporangium, there are individual spores. Here's a side view of a sorus. Here's a sorus. Here are the individual sporangia with spores inside. And this is the leaflet here. So again, the leaflet is part of the sporophyte plant. Sorus, cluster of sporangia with spores inside. And then as we go up on higher power, again, this is an individual sporangium with haploid spores within. And so what will happen then as the plant matures, the sori develop, the sporangia develop, the spores mature within, and then the sporangia will rupture, releasing the spores, the haploid spores. The haploid spores, if conditions are favorable, will begin to mitotically divide, and the spores will grow into a gametophyte plant. And in the case of the ferns then, this is a separate plant. The gametophyte is only about, a 1/16 of an inch, an 1/8 of an inch, maybe as big of a 1/4 of an inch in some species, but it's small, and it's a separate plant. And it would be living on its own, a free living gametophyte, and that would be a haploid plant, or is a haploid plant. So they're usually kind of heart-shaped like this. And so, again, the role of the gametophyte is to make gametes. And so to get there first, the spores have to germinate. So again, we would be looking at protonymata. Protonymata are germinating spores. The spores grow into gametophytes. And then as we talked about before, the gametophytes will produce antheridia. So again, as we look at the entire gametophyte here, these are the antheridia. The little specks inside are the spermatozoa. When those rupture, the spermatozoa are released, and so they have to swim to find an ovum. And so it depends on the species, but typically on a separate gametophyte, a female gametophyte, it will produce archegonia. And so those archegonia will produce ova, just like we talked about with the mosses. And so here we're looking at a top view of the archegonia, so we're looking right down the top, so it's kind of similar to what we talked about before, a vase-like structure. And so the sperm are going to have to swim right down the top of that. But again, the important thing is that water is required here because the sperm have to swim to the archegonia and swim down the top. And then as we can see they swim down the top. There will be an ovum in there that they can fertilize. And when they fertilize that ovum, it's no longer an ovum, it's a diploid zygote. And so now we're back into the sporophyte generation, the diploid generation. So again, Antheridia will produce sperm, Archegonia will produce eggs. Sperm swim down the top of the archegonium, fertilize the egg. That forms a zygote. And so now the zygote, again, if conditions are favorable, will begin to mitotically divide and begin to grow into a new sporophyte. And so that's what we're looking at here. And so literally then, as this young sporophyte begins to grow, this is where an archegonium was previously. So again, there was an archegonium with an ovum that was fertilized, that formed a zygote. Zygote begins to mitotically divide. And so now this is the new young diploid sporophyte fern growing out of what was a archegonium. So we can see the beginning of the vascular tissues already beginning to form here. Leaflets beginning to form or initially. So this is a young sporophyte. And so that sporophyte, as it grows, is obviously just going to obliterate what was the gametophyte. So again, the gametophyte is just a tiny, tiny structure. It's been fertilized. The sporophyte begins to grow, and it will rip apart that gametophyte as it grows. And then it will mature eventually into a fern, and that's how the next generation of ferns is produced. So there are a number of different ferns that we find in the Yellowstone ecosystem. Examples include the bracken fern. That's probably the most common fern that we see up there. And one thing I do want to mention is that as ferns grow, the fronds grow in what we call fiddleheads. So these young ferns are kind of unfurling. We refer to those as fiddleheads. Some species of fern actually are edible. Some people eat fiddleheads. And then some other types of ferns that we see, parsley fern, cliff fern, among many others. So ferns, even though they're primitive in terms of their life cycle, they are a very, very successful type of plant. But the problem for them in an evolutionary sense when they first appeared is that as Earth got drier and also as drier habitats were established, they really could not invade those dry habitats. So they do have true roots that can tap into water underground, but they're still tied to wet or humid habitats because they need water for fertilization. They are not seed producers. Okay, that'll do it for today. I'll see you next time.